So our webinar on the systematic approach for the evaluation, validation and implementation of NAT-based Mycoplasma detection methods. We have 24 colleagues on the call, but a lot more have registered, so up to 80. And I guess these colleagues will then watch the recording afterwards. So, well, welcome to everybody who is here now. Everybody will receive the recording in a few days. We can't guarantee in the holiday period how quickly it will be sent out. It might take up to 2 weeks, but you will definitely get it. So if you continue to the next slide. Thank you. So this webinar is held by BioPhorum on behalf of the Rapid Methods for Adventitious Agents teams, and 2 colleagues have prepared today's presentations. So before we start, I would just like to say a few words about BioPhorum for those of you who are not familiar with BioPhorum. Next slide, please. So BioPhorum is an organization that provides a platform for the biopharmaceutical companies to collaborate, share and pool knowledge and build solutions together that in the end benefit the industry as a whole. BioPhorum is organized in several forums, hence the name BioPhorum, and within each forum we have various work streams. Each work stream itself is led by a dedicated facilitator. I'm the facilitator for Rapid Methods for Adventitious Agents team in the Technology Strategy Forum. And the team has published 2 papers that will form the basis for today's presentation. Next slide, please. So, as you will all know, there are several alternative and rapid micro methods that are available to detect microbiological contaminations, and regulatory authorities are encouraging the biopharmaceutical industry to adopt these innovative methods. To help with that, the ARMM team within Fill Finish published a paper in 2020 which provided a framework for the evaluation, validation and implementation of these new methods. And this framework was then adapted by the Rapid Methods for Adventitious Agents teams and published in June 2022, and a subsequent paper with case studies was then published in February this year. And this is what this webinar is about. Next slide, please. Bit of housekeeping before we start. You will have noticed that you are all on mute already and that your cameras are switched off. And this is for the first part of the webinar, i.e. the presentation. Then we will have the Q&A session afterwards, and for that you will be able to switch your camera on and also, of course, you know, unmute yourself, or you will be unmuted rather. Yes, so as I said before, after the webinar you will receive a link to the recording, and during the presentation please put in any question that comes to mind into the chat, and we will go through the questions then in the Q&A session. Next slide, please. And the next one. So we've got 2 experts today who have been very much involved and were, very, very important members for our team and have been very much involved in writing the paper. So first of all, we've got Kevin McCarthy from Roche Genentech, and then Michael Brewer from Thermo Fisher Scientific. And I will not go through their impressive bio now, but will hand over to Kevin to start with the content straight away. Thank you.
Okay, thanks, Margaret. I'm gonna go ahead and take control, Nettie. Okay, hopefully I didn't turn it off for everybody. Hopefully everybody can still see.
We can.
So first of all, my name is Kevin McCarthy. I'm a global QC scientist with Roche. And in my section, I'm going to present some of the generalities of a 9-step process that our BioPhorum group agreed to. And then I'll also introduce the first of 2 case studies from the well, one from Roche and one from Thermo. So, whoops, I just messed up my presentation. Okay. So there's several alternative there are several new alternative and rapid microbial methods available for detecting and enumerating microorganisms, including adventitious agents, and that's a no-brainer. We all know this. Regulatory, as Margaret said, regulatory authorities are encouraging industry to adopt these new and innovative technologies. And as many of us have experienced over the years, is that when we implement a new technology, it ends up being that there's a lot of questions asked having to do with, well, this company did it this way and another company did it a different way. So the regulatory authorities come back and they're asking for a lot more information than is typically given for a dossier filing. So what we've decided is we came up with the BioPhorum arbitrated our company's discussion about the best way to tackle this. The idea being that these methods will lead to improved monitoring and quality assurance of biopharmaceutical processes and manufacturing environments with shortened time to release. So our group came up with a general 9-step framework to describe the approach and common language for implementation of the various new methods that are available to us. In addition to today's case studies, the approach has been used for projects such as alternative and rapid sterility tests and automated colony counters. The general framework has 9 steps, and to break it down briefly at this point is step 1 is to identify an operational or business need. 2 is define the application. 3 is assess the requirements. 4 is compare the options and technologies. And 5 is develop a business case. 6 is to perform your proof of concept studies. Then step 7, you validate at a pilot or primary site. And 8 is you deploy it globally or company-wide, depending on your situation. And then step 9 is review considerations for regulatory filings and implementation strategy. Okay, next slide. All right, there are variations in the approach taken by various biopharmaceutical manufacturers to implement alternative nucleic acid testing based adventitious agents methods. The general ARMM framework paper has been adapted to address the need for a systemic and best practice approach to evaluating, validating, and implementing new alternative adventitious agents methods. Next slide. Okay. So the nucleic acid mycoplasma detection method, the suggested 9-step process for the initial validation to final implementation of the method has been applied in a follow-up paper to nucleic acid testing based mycoplasma detection. And listed to the right on this slide is a list of the participating companies that participated in it. And Margaret, I'm not sure if you see this, but my slide is showing it very blurry, the names of the companies.
Well, I can read it, so
okay.
hopefully everybody else can.
All right, so then I'll continue. So in addition to the 9-step framework that we established, we also established a common URS that the companies participated in developing. And this, of course, will facilitate step 3 of that 9-step process. The URS that we decided on had 4 overall sections with varying amounts of criteria. The first section is a system compatibility requirements that had 6 criteria that we identified. The next one is the network security requirements with 2 identified criteria. Then we had the software security requirements with 6 criteria, and then the 21 CFR Part 11 with 24 criteria, 7 of which had to deal with electronic signature requirements. Okay, and again, the 9-step approach is to reiterate, is to identify an operational business need. In the case of, and this will be for Roche, we wanted to reduce the lead time, define the application. For Mycoplasma, we wanted qualitative release testing, identify the requirements. So we're using real-time PCR, and the time to result release time validated and equivalent and compendial method. Validated and equivalent to compendial methods is all covered in the URS. And then for the business case development, there's a reduced time to release to patient, which is important from a business standpoint. Steps 4, 6, 7, 8, and 9, I'm going to go in depth in the following slides. Okay. For the Roche case study, it's called the implementation was a system called MycoTool, which is a Mycoplasma real-time PCR analysis in a kit form from Roche. Okay, and as I mentioned, going back to step 4 on that 9-step process, the technology background and comparison to other options that we had. So we started the MycoTool development started 10 years ago, 10 to 15 years ago. So it's been well hashed out. So the Roche MycoTool is a real-time PCR-based method for detecting mycoplasma in CHO cell matrix. And of course, the sample type is our CHO cell PHCCF, or pre-harvest cell culture fluid, with a sample volume of 1 ml. The first section of the method is the DNA extraction, which is accomplished on an automated Mycopure 96 extraction instrument from Roche. This is a magnetic bead-based technology in a 96-well format. The extracted DNA is used in a real-time PCR assay that's run on a light cycler 480, again from Roche. The PCR is done using 2 fluorescent channels that maximize, I'm trying to point with my pointer, that maximize the ability to detect different signals. The assay has several built-in controls, including a positive control, a negative control, an internal control, and a NC and a negative control CHO cell sample that's spiked with an internal control as well. And that's the extraction control. And these are the instrument. This is the system at a high level. The first one is the Mycopure 96. The second instrument is the light cycler 480. And then the Mycopure 96 has we'll say kits or reagents that come in kits that are well qualified and QC controlled. And then the light cycler 480 PCR system also uses the MycoTool Mycoplasma real-time PCR kit, which is also quality controlled and contains all of the required reagents for the assay. So let's go on to step 6, which is proof of concept. So we started out using the 16S DNA gene and aligning the sequences for Mycoplasma hominis, orale, arginini, A. coli plasma leidlaui, Mycoplasma hyarinus, pneumoniae salivarium fermentans, and Spiroplasma citrate. So the primers and probes derived from the above alignments were submitted to BLAST database and determine what other Mollicutus strains we could possibly detect with the primers and probes. Okay, so we took dilutions of the above mentioned strain, just those ones. Sorry about that. Just the ones that we identified in the first bullet comment. So we took dilutions of those strains and did initial extraction and real-time PCR development and optimization using the extracted DNA. Later development of the method included addressing the need to producing the assay as a marketed kit. A kit configuration allows for more efficient quality control and reagents and quality control of all the reagents simultaneously. Okay, and let me go back to this one. So the proof of concept, basically, we proved that it works, but that's a very tiny bit of the puzzle. The next step is the validation, and that's more intense. So per the requirements of EP 6.2.7, the validation included sensitivity, or LOD, with a maximum acceptance criteria at 10 CFU per mil. Specificity, the initial generic validation was conducted using the species that we've already mentioned: hominis, orale, arginini, leidlaui, hyarinus, pneumoniae salivarium fermentans, and citrate. The robustness included precision and cross-contamination assessments. We did comparability to the compendial method was also executed. 3 different lots were used for the validation, and each lot is aliquoted and spiked with a dilution of each of the specified test species. Each dilution was in turn tested 8 times per lot, giving 24 data points for each dilution point for each species. The validated statistics program PROBIT was used to calculate the LODs. Okay, so specificity. The specificity of the assay was determined with Strep bovis and Lactobacillus acidophilus and Clostridium sporanges. The detection of each of the 3 bacteria was examined in 3 independent dilution series in 8-fold determinations. And the idea for that is again to get 24 data points. Gram-positive bacteria were spiked into CHO cells using different titers, 10 to the 2nd and 10 to the 6th CFU per mil in the Mycoplasma PCR. No acceptance criteria was set with regard to the cross detection of the gram-positive bacteria. In addition to development based on the identified Mycoplasma species, blast analysis was performed with the primers and probes using Primer 3 calculation software. And the results of that were we were able to identify 125 Mycoplasma species, 10 A. coli plasma species, and 9 Spiroplasma and Uroplasma species that should be able to be identified with the primers and probes that we developed. Step 7 for validation, we're talking about robustness now. The robustness was tested with 10 CHO cell samples spiked with M. orale at a concentration of 3 times the calculated LOD, which is about 1.5 CFU per mil. Additionally, 2 batches of mycotool mycoplasma kits were used. The 10 spike samples must be found to be positive. The mean of the detected CP values is calculated, and it must not exceed a range of plus or minus 4 CP values. Okay, then we looked at the precision, and the precision was examined on 3 separate dilution series in an 8-fold determination, again, to give us 24 data points. In addition, the CHO cells were spiked with Mycoplasma orale and with the Mycoplasma orale and investigated by the real-time PCR assay. The concentration of Mycoplasma orale was used at 3 times the calculated LOD. The acceptance criteria were met, and the mycoplasma were judged to be positive for each analyst, and the mean of all the detected CP values were within the plus or minus 4 CP range. We looked at cross-contamination also, and the cross-contamination was done using 10 unspiked CHO cell samples and 10 CHO samples spiked with M. orale at a concentration, and the concentration was 100 times the limit of detection, or 50 CFU per mil. The non-spiked and the spiked samples were loaded alternatively, and all 10 spiked M. orale samples were found to be positive, and all of the unspiked were negative. And we also looked at the at validation comparability. We did side-by-side testing with the culture method and the indicator culture method as well. Testing was done using NC CHO cells spiked with 10-fold dilutions of the target species, and the concentrations were at 100 CFU and down to 0.001 CFU per mil. So validation sum, and again, the step 7, the validation step, to me was the most important and the most intricate. But to summarize the validation, so the robustness, precision, and cross-contamination, Mycoplasma CCR method is unaffected by small but deliberate variations using different Magnapure 96 and Light Cycle 480 systems, using different commercial lots of the Myco toolkit, and a test performed on different days using different analysts. This for the specificity, 150 mycoplasma species were determined to be able to be detected, and this was determined through sequence alignment. Slight gap in specificity with gram-positive bacteria. This slight gap in specificity is not relevant for biological production, since additional required methods such as bioburden testing or the test for absence of bacteria and fungi would detect any bacterial or fungal contamination and reject the lot anyway. The sensitivity and detection limit fulfills the requirements according to the European Pharmacopoeia 2.6.7 for the replacement of the culture and indicator cell method. If the alternative NAT is proposed to replace the culture method, the NAT must have a detection limit of 10 CFU per mil, which they have so far for Roche. Okay. So now we'll move on to step 8 of the 9-step process, which is global deployment. So after the initial development and optimization, we established a training program where the analysts would travel to this site where the assay was developed and take training there. After all sites had been trained and the required instrumentation installed and validated, each site, and this is an important section that saves a lot of time and money, each site acted as an additional, went back to their home campuses, and each campus acted as an additional lab and analyst for the robustness testing and the validation. And what this did is it kind of made it a co-validation where it could be the analysts at different sites can participate in the validation, and therefore it would be also considered a method transfer as well. After the validation/transfer, 3 centers of excellence were established to conduct network testing based on regional production locations. Each lab could also act as a backup lab to the others. Currently, we have a lab in Pennsburg, Germany, Oceanside, California, and South San Francisco, California. The initial validation and subsequent 5 product validations were full validations, which used all of the strains, all the molecular strains identified previously. Due to the reliability and repeatability of the validations, however, we decided to make subsequent product validations abbreviated such that they only employed Mycoplasma hyerinus and Mycoplasma fermentans to establish the LOD. Okay, so for our regulatory landscape, currently the MycoPool tool has been validated for all Roche commercial products and filed to act as a valid replacement for the compendial methods. Acceptance by the health authorities is of course piecemeal, but the Mycoplasma has been accepted for at least 25 commercial products. Let me see, next slide. So additionally, currently Roche is employing this 9-step strategy as well for assessing a flow cytometry system. And I think that concludes my section, my case study presentation. And now I will hand it off to Mike Brewer to present Thermo Fisher's case study. Mike, you want to take it?
Yep, thanks, Kevin. Okay, I've got control now. So this is Mike Brewer from Thermo Fisher Scientific, and I'm going to talk to you today about an example of validation of the MycoSeq Mycoplasma detection assay. As part of that, I'll share some background on the development and the attributes of that method, and then share results of a typical validation study. So the guidance that we applied in the design of this validation study first followed the European Pharmacopoeia 2.6.7 published in 2007, both for sensitivity at 10 genome copy per mil for purified DNA, and then for the live mycoplasma arm of the study, 10 CFU per mil for mycoplasma cells. And these were carefully prepared and evaluated to ensure that the genome copy to CFU ratio was close to 1-to-1 as possible. Then we also applied ICH Q2R1 guidance of validation of analytical procedures, texts, and methodologies. Specifically, it's a limit test for impurities, impurity being Mycoplasma DNA, whether it was recovered from pure DNA spikes or live Mycoplasma spiked into the sample. The LOD that we validated was 10 genome copy per mil for purified DNA, 10 CFU per mil for Mycoplasma cells. We also assess specificity, and that's in two parts. No detection of off-target species was confirmed, and then detection of Mycoplasma DNA at the LOD following extraction from the CHO cell culture. So showing that components in the typical test sample did not interfere with detection at LOD. And finally, we also incorporated feedback from a discussion that took place at the FDA PDA workshop, Rapid Methods for Detecting Mycoplasma Contamination, that was held back in September 23 of 2008. And that discussion really centered on using a sufficiently large and representative test sample volume, that is 10 mils. So that's the starting test sample volume we selected for this study. Just a little background on the MycoSeq assay. It was designed for lot release testing in GMP environment, specifically designed to meet or exceed the EP guidance from 2007. It employs an optimized sample preparation protocol for lot release testing applications. It also uses an integrated software analysis platform to enable not only automated positive-negative calls, but also enables full 21 CFR Part 11 compliance. As part of that, we also, to fully support our system, we offer instrument validation services, IQ/OQ, and then global support network of experienced field application scientists that can provide full workflow training from sample prep to results interpretation. The performance of the assay, it's sensitive and enables validation at levels of 10 genome copies per mil or below, or 10 CFU per mil if you're using live Mycoplasma. That's specific. There's no detection of off-target organisms. It employs objective multi-parameter results interpretation, which I will cover the details, and it has full workflow controls, including a patented discriminatory positive control for the highest confidence in both positive and negative test results. Following validation, regulatory acceptances is an important consideration for anybody considering a kit. MycoSeq, following validation, regulatory filing, and review, our customers have received regulatory acceptance to use MycoSeq for lot release testing applications across multiple therapeutic modalities. This includes cell culture manufacturing, cell therapy, tissue therapy, cell-based therapeutics, as well as vaccines. And there are validations in process for producing gene therapies, or specifically recombinant AAV. It's like the Myco tool, we offer a complete sample to answer solution, starting with sample prep, also based on magnetic bead technology. Most of our customers use an instrument called the Automate Express, which automatically extracts the nucleic acid from the test samples. The qPCR detection assay, the qPCR instruments, and finally the AccuSeq software. When we talk about these high sensitivity assays, there's really three components that go into it. First component is treatment of the sample background reduction, and then Mycoplasma contamination. That's accomplished by centrifugation. Again, in this study, we're going to talk about 10 mil samples of CHO cells, and then these were approximately 10 to the 6 cells per mil. Following extraction and elution, 1 mil equivalent of the test sample is evaluated in each qPCR reaction. Following the background reduction, we extract the nucleic acid. This uses a very high percentage magnetic bead-based extraction kit. It enables very high % nucleic acid recovery. We actually share it with our quantitative host cell DNA assay and typically get yields of or recoveries in the 80 to 100% range from complex matrices. And then finally, it's followed by detection of the Mycoplasma DNA using the highest sensitivity qPCR assay. I say that because it's based on cyber green detection chemistry, which I'll describe briefly. That it is sensitive to the level of 1 to 3 copies of target per qPCR reaction. The MycoSeq assay is a highly multiplex, multiple primer based design. Uses over 40 Mycoplasma specific primers in a single reaction to enable the coverage of over 140 Mycoplasma species. And importantly, it doesn't cross-react with any bacterial or non-Mycoplasma species. Now, all this adds up to high sensitivity Mycoplasma detection. Because it's cyber green based detection chemistry, we can use 3 parameters for results interpretation. First one is the cyclic threshold. That's the qPCR cycle where it crosses the fluorescence level reaches a threshold. This is a measure of the target DNA level at the beginning of the PCR reaction. The second parameter the second and third parameters we use are analysis of the melting of the amplicon. Detection based on cyber green dye. The cyber green dye actually intercalates into double-stranded DNA, and it becomes fluorescent when it's bound to double-stranded DNA. So to analyze the amplicons, we can melt that DNA and look at the temperature in which it melts and also the amount of fluorescence that's released. So the derivative value is a measure of the specific amplicon created during the PCR reaction. And that's indicated by the peak height, the amount of fluorescence released during melt. And then the melting temperature. This is very important because we understand, that based on our development work, that the melting temperature of Mycoplasma amplicons falls between 75 and 81 degrees. And here's what the actually raw data looks like. This is with the assay. This is analysis of a 10-fold dilution series of purified Mycoplasma arginine DNA. It goes from 100,000 copies here at the lower CT and then at the higher CT level. It shows detection of 1 copy of Mycoplasma arginine DNA per qPCR reaction. And then followed by the melt analysis of the amplicons created at the 1 genome copies per qPCR reaction level. I also want to cover an important feature of the assay, and this was actually developed due to some lessons I learned early in my career in implementation of highly sensitive qPCR assays. So one thing I found is if you accidentally contaminated your test reaction with control, it could lead to a false positive or false positive result or an invalid result. So we specifically designed a discriminatory positive extraction control with an amplicon or a target of the assay that's amplified by the Mycoplasma specific primers, but it generates an amplicon that's outside the range of Mycoplasma amplicons, as you can see here. And in this example, a melting temperature of about 80 degrees for Mycoplasma and 85 degrees here for the discriminatory positive control amplicon. So this enables, you can analyze all positive results and make sure that the target or the signal is being generated by Mycoplasma, not control. It's also important with these assays to understand the analyte, and that is the relationship between a colony forming unit, a CFU, and a genome copy, which is the target of the assay. So we illustrate a Mycoplasma cell in this cartoon, and inside that Mycoplasma cell is the Mycoplasma chromosome, which is circular, and there's 1 circular chromosome or genome copy per cell. Our assay, just like MycoTool, targets a conserved region across the species. It's a 16S ribosomal RNA gene. And what's interesting about Mycoplasma is some species have one copy of this target gene, other species have two copies. That's important only in that for the species with two copies of the target gene, you get a little bit increased sensitivity for those species at the genome copy level. But ideally, when you're using live Mycoplasma stocks, you should have stocks that one genome copy equals one CFU. But that should be verified, and I'll show you some data on how that can be done. But if that ratio changes because the sample or stock has cells that contain DNA but do not grow or represent as a CFU, you can get a misleading indication of the sensitivity of your nucleic acid test, because that's what the qPCR assay is measuring. And here's some data that was published by the FDA research team back in 2011 that speaks exactly to that phenomenon. And what they did here is looked at Mycoplasma cultures during growth and measuring both genome copies in the diamonds here, and then in the squares they're looking at plate culture, counting the CFU. And then in the diamonds represent the genome copy to CFU ratio. And what you can see in early stage growth, the Mycoplasma, they both track quite in line with each other, one CFU, one genome copy. But at some point, the Mycoplasma cells reach a stationary phase and then enter a death phase. And in that, when that happens, the CFU levels go down, the DNA levels stay about the same. And then you can see down here with the diamonds, you're tracking an increase in the genome copy to CFU ratio. So, the point of this study was to teach people and illustrate why it's very important with live Mycoplasma stocks to use them early in log phase growth, where the ratio of genome copy to CFU is very close to 1-to-1. Otherwise, you can get a misleading result. Now I'll go into the example validation study plan. As I said earlier, we're covering two aspects per ICHQ2, specificity, which part one of specificity is no detection of non-Mycoplasma bacteria. Also, we have and this data is contained in a drug master file that we have in place with both CBER and CDER, as well as Health Canada. Second is no interference from the test sample matrices, and that's really addressed in your spike recovery at LOD. So LOD part one of the study, this was for each Mycoplasma species included in the study, 6 samples with 100 copies or 10 GC per mil purified Mycoplasma DNA spiked into a lysate from the 10 mil samples, 4 PCR reactions per extraction for a total of 24 PCR results. For LOD part 2, and our early adopters actually validated just like this, used both pure DNA as well as live Mycoplasma. Now, not as many people use live Mycoplasma for a variety of reasons, which I'll discuss in a minute. But for part 2, these were 6 samples spiked with 100 CFU or 10 CFU per mil of live Mycoplasma cells spiked directly into the 10 mil cultures, recovered, and the DNA extracted. And again, the analysis went as the same of 4 reactions per extraction for 24 total PCR reactions. Here's a summary of the specificity results. We looked at both host cell DNAs as well as genetically related bacterial and fungal species. And you can see here hamster, human, and mouse DNA, as well as Bacillus cereus, subtilis, and a selected set of organisms that bioinformatically were most closely related to the molecule species. The important data is really here in column 4 of each of the triplicate reactions, and that is none of these species were detected at 10,000 genome copies per reaction. Second part, and this is the real important part, is the LOD. This is section 1, purified mycoplasma DNA. Again, purified DNA was spiked into a lysate created from a 10 ml sample of the CHO cells. We evaluated 10 species as the 8 species as indicated or recommended in the European Pharmacopoeia, as well as 2 other mycoplasma species that could be related, or human-borne mycoplasma, which we felt would be important to include in the study. In this case, and again, here in column 3, 100% of the 24 reactions were positive for mycoplasma. These are the CT values, which are important. One, because you can use these CT values for comparison between individual studies, and also as part of that genome copy to CFU assessment if you need to do that. In this case, of course, this is just genome copies. We also did some statistical analysis because, as many of you know, as you approach the lower limit of detection with any analytical test, you start to get a high degree of variability. What you can see here in this analysis of the CT results is very, very consistent across the 24 results, indicating that you're not approaching the lowest limit of detection capability of the method. Again, here is part 2 of the case study. This was live mycoplasma. This we did, it was a lower number of samples, but total number of tests, total positive reactions, again, 100% positive. And the important thing here is looking at the CT values. You can see that indeed these were mycoplasma stocks that were carefully selected for low GC-CFU ratio, and the CT values indicate that there was not an excess of genome copies present in the live mycoplasma arm of the study. Just want to talk a little bit more about this, and this relates to the document that Kevin and I are basing our webinar off of today. The team did include a rationale supporting the use of purified mycoplasma DNA for validation of qPCR or NAT-based detection methods, as long as the NAT that's targeted by the method is DNA. But the analyte, the rationale here, the analyte is detected with a qPCR or a DNA-based detection method is mycoplasma genomic DNA. It's not a cell or colony-forming unit. But of course, if a cell is there and the genome is extracted, it is detected. It's been well established that mycoplasma genomic DNA can be purified and accurately quantitated, often using orthogonal methods. Stability of purified DNA has been demonstrated. And as I showed you before, that was just a summary of it, but preparation of accurately quantitated mycoplasma stocks can present challenges that are not limited to variability of genome copy to CFU, accuracy of CFU titer due to clumping or aggregation of some species, and these can lead to misleading results. And also, it's been shown that the stability of mycoplasma stocks are difficult to maintain. Degradation can lead to misleading results in validation or even validation failures if the stocks have degraded. And then this is very important in many testing laboratories. Use of live mycoplasma is not allowed because of unacceptable risk. And finally, concentration of mycoplasma by centrifugation, that's been well established. Here's another, this is just an example. I wanted to include this to show how you can use the CT results to compare results from lab to lab, study to study, or if it's mycoplasma, either live mycoplasma or purified DNA. The top 2 frames are the results from the study that I just presented. And then the bottom 2 frames are results from a customer validation study that performed their study, very similar to how I described it, with both an arm of pure DNA and an arm of live organisms. They chose to be a little more conservative on the LOD, so they selected an LOD of 8 genome copies per mil or 8 CFU per mil. But what you can see here is, 100% positive in both studies, and then importantly, a CT value, right around 31 as the CT. So very, very consistent from site to site, different Mycoplasma DNA and CFU stocks prepared, but very, very similar and comparable data between the two labs. And finally, looking at, when you look at that CT value on a standard curve, prepared by analysis of a dilution series of purified Mycoplasma arginini DNA, what you see here is that CT of 31 that was reported in these validation studies is very, very consistent with the CT of 31 that you would expect for 10 copies per reaction. And finally, early on, and some regulators still continue to ask, like, okay, we understand you validated at 10 genome copies or CFU per mil. What's the lower limit of detection for the species that you evaluated? And with qPCR, you can actually do this. Because for efficient linear qPCR assays, a 1 CT difference represents a twofold difference in the starting quantity of the target DNA. So as an example here, using that Mycoplasma arginini results from those two validation studies, the CT of 31 for 10 copies, that would correlate to CT of 32 for 5 copies, 2 to 3 copies would be 33, and down on the 1 copy would be about 34. When you look at that standard curve again, this calculation is actually consistent with the real results obtained. So that's an easy way to estimate lower limit of detection by using principles of qPCR as opposed to using a statistical tool. Robustness, and that can be addressed per ICH, can be addressed in method development. We extensively evaluated the robustness of the method, and that data is detailed in the drug master file. But many of our customers include user-specific robustness options, and a couple of examples of that is test sample storage conditions following extraction, and evaluating multiple conditions, and also storage or test sample storage as well as storage of extracted DNA. So those are 2 examples of robustness testing. So in summary, the assay is sensitive. It's specific for Mycoplasma. The results interpretation uses 3 parameters, the control, and then the support we offer. And also say, our early adopters did execute comparability to the 28-day test, and comparability testing of identical sample spike with 10 CFU of Mycoplasma DNA. And the summary of that, and about 80 samples tested in comparability, it turned out that, and this comparability was done at 10 CFU per mil or less, but in that, 38% of those samples that contained enough Mycoplasma to be detected by either method, 38% of them failed to detect Mycoplasma with the compendial test, but 100% of them were detected by the MycoSeq assay.